You're listening to Conferences Online Allergy from Children's Mercy Hospital in Kansas City, Missouri. Today is November 27, 2023. We will be doing Abbas Chapter 1, Properties and Overview of Immune System, presented by Dr. Nikita Raje. She's an Associate Professor of Internal Medicine and Pediatrics and is the Chief in the Section of Allergy Immunology at Children's Mercy Hospital.
We are going to switch gears today, move from Lawrence Somparak's textbook to Abbas, Littman, and Pillai's textbook called Cellular and Molecular Immunology. And we'll start with the chapter 1, which is Properties and Overview of Immune Responses. Hope you guys got some time to go through this chapter. Feel free to stop me if you have questions in between. Again, we'll be talking about each chapter as we go through this textbook. So hopefully the recording is on. It looks like you can hear me well and see the slides. So let's get started. I'm Nikita Raje from Children's Mercy, University of Missouri, Kansas City. Today is November 27, 2023. Let's talk about Abbas Chapter 1. Here are my disclosures, nothing relevant for the topic today. Our learning objectives for today are to recognize the types of immunity, to differentiate between innate and adaptive immunity, and to recognize the role of humoral and cell-mediated immunity. Right, so we'll be talking about different this is basically the outline, and we'll be talking about some of these topics. We'll come back to that. Here are a couple of pre-test questions, and we'll come back to the answers at the end of the chapter. So before I forget, let me tell you, this is supposed to be a short chapter. We'll see how it goes. If we do have time at the end of the chapter, we'll go through a Jeopardy for Somparak textbook, which we had skipped because of the scheduling. So let's get started with these questions. Which of the statements is true? Phagocytosed microbes are killed by antibodies. Intracellular microbes are killed by humoral immunity. Antibodies can eliminate the reservoirs of viral infections, and extracellular microbes may need cell-mediated and humoral immunity. Question 2, which of the following is a distinct feature of innate immunity? Diversity, memory, non-reactivity to self, and specificity to individual microbe. All right, so getting started with the discussion. So what is immune response? Immune response is a reaction to the components of microbes as well as to molecules that are recognized as foreign, regardless of the physiological or pathological consequences of such a reaction. So it doesn't matter at the end of that reaction, after the immune response is produced, whether there is a pathological consequence or physiological immune response that is helpful to the body or not. So we know there are two types of immunity, innate and adaptive immunity. And innate immunity is natural and natural or native immunity, which means it's hardwired and it's already in place before the infection occurs. It's capable of producing the response pretty quickly, and it stays the same with repeated exposure. The immune response is to specific structures that are common to a group of related microbes in terms of innate immunity, compared to adaptive immunity, where it is stimulated after exposure to the microbe, and there is increase in magnitude and capability with each successive exposure. It is specific for various structures, even in the same microbe, and it's only found in vertebrates. So we'll go through some of these components of each type of immunity in a little bit. Let's look at the characteristics before that. So we know that the specificity for each of these immune systems is a little different. So for innate immunity, as I mentioned, the specificity is for the shared group of patterns that are noted in microbes, various microbes, or molecules that are produced by damaged host cells, compared to this adaptive immunity, which is very specific for microbial or non-microbial antigens. And because of this, the response may be more pronounced or more sophisticated in terms of the adaptive immune response. Innate immune system can recognize molecules that are encoded by the germline genes, and which means that there is limited diversity compared to adaptive immune system, which undergoes somatic recombination of gene segments in lymphocytes, and that gives rise to large diversity. We know that innate immunity also has some memory, but it is pretty limited compared to adaptive immune system, which has good memory of what the response was to previous infection. But one common feature for both is they can recognize self from non-self. And that's really important because we want to make sure that the immune response that's produced in a human body is only to foreign molecules and the ones that are dangerous, and not to self molecules. So it's the same for the innate versus adaptive immune system, although the mechanisms may be different. And of course, the components of each of these systems vary. So let's start with the components of innate immunity. So the first line of defense is provided by a physical barrier, and that physical barrier is provided by skin, mucous membrane, and also some chemicals in those physical barriers, and that's provided by antimicrobial chemicals. There are certain cells of the innate immune system, such as phagocytes, neutrophils, and macrophages, other cells like eosinophils and basophils, natural killer cells or other innate lymphoid cells or ILCs, mast cells, dendritic cells. All these cells provide immune response, and these cells are ready to go. They are in place before the infection occurs, and they can assemble their weapons pretty quickly after the infection starts. There are some proteins that take part in providing innate response, which are complement proteins, and some others that we'll be talking about over a period of time. When it comes to adaptive immune response, there are a couple of types of adaptive immune responses, and then we'll talk about active and passive immunity as well. So in terms of adaptive immune response, it's provided by either humoral immunity or cell-mediated immunity. The humoral immunity is against microbes and their toxins that are outside of cells, so extracellular microbes. Outside of cells means maybe the lumen of GI tract or respiratory tract or in the blood itself, which means that these microbes are outside of the cell linings, and so they need specific mechanisms that are able to reach those areas. We know that the humoral immunity is provided by B lymphocytes that produce antibodies, and that helps in blocking infections and eliminating these extracellular microbes. Now, when we compare this to cell-mediated immunity has, again, two different types, right? One that's against the extracellular microbes, and the other one that's against intracellular microbes. So the extracellular microbes are dealt with by helper T cells, whereas intracellular microbes are dealt with by cytotoxic T lymphocytes. So the T cells, the cell-mediated immunity is provided by these T cells in two different forms. The helper T cells help in producing some molecules called cytokines that make sure that the cells needed to deal with these extracellular microbes are activated, and they have enough weapons to kill those microbes, such as macrophages or neutrophils. So the activated phagocytes are the ones that kill microbes, but these phagocytes are enabled by these helper T cells. And they become more efficient in presence of these helper T cells, compared to cytotoxic T cells, which kill the infected cells and eliminate the reservoirs of infection of these intracellular microbes such as viruses. So we talked about three different lymphocytes already: B cells that produce antibodies, and these antibodies help in neutralization of microbes, help in phagocytosis and complement activation. Whereas helper T cells that we looked at produce these molecules called cytokines, and they help these cytokines will determine what kind of cell it can help. For example, it can help in activation of macrophages, it can produce inflammation, it can activate other B and T cells, right? So all of these cytokines can differ based on what kind of infection is being dealt with. Cytotoxic T cells, we saw that it can kill the reservoirs of infection. So any kind of infected cell or damaged cell is what they're gonna go after. And then there are cells called regulatory T cells. Regulatory T cells, once they come across the antigen, which might be, say, a self antigen, they are going to actually suppress lymphocytes from proliferating, forming clones, and producing immune response so that they can protect against immune response against self antigens. Also, adaptive immune response can be present in two different forms: active and passive immunity. Active immunity is what we just talked about. Say a microbe or microbial antigen enters the body, say in form of infection or in form of vaccine. And over a period of time, those T and B lymphocytes are going to proliferate, get activated, proliferate, and differentiate to produce those weapons that are used to fight those infection or provide immune response to the vaccine antigen. This takes days to weeks for this immune response to be produced. Once that immune response is produced, the infection is going to clear and there is recovery from that infection. Compared to passive immunity, where, say, antibodies are administered directly to a particular infection. So that might not be able to adapt over time. It's just that there is administration of the antibodies given to an individual, because of which it will protect that person against that infection and provide immunity and the immune response. The difference between these two types of immunity is that the memory is produced from active immunity because of production of memory T cells. However, in terms of passive immunity, there is no production of memory. Both of them are specific to the same infection, but the memory is only produced by active immunity. All right, a few definitions. What is an antigen? It's a substance that binds to specific lymphocyte receptor. When you think about antigen, well, that's pretty straightforward. But when you think of immunogen, how is it different from antigen? So to keep in mind that any substance that can bind to the specific lymphocyte receptor is called antigen. But the substances, after binding to that lymphocyte receptor, produce an immune response are called immunogens. And what is opsonization? I believe we talked about this in Somparak as well. Opsonization is a process in which some factors in serum enhance phagocytosis by coating the microbes. So they pretty much tag the microbe for destruction, and that they can be eaten up by the phagocytes pretty easily because they are tagged, where it basically is a label to say, here's a sign that the phagocytes can follow and take those up and kill those microbes. That's called opsonization. All right. Now let's talk about the cardinal features of adaptive immune responses. So we looked at that chart that gave those cardinal features or characteristics, and we are going to go into the details of what exactly each of these mean. So for the first one is specificity. So the response has to be specific to a particular antigen. It could be for distinct antigens, the response has to be specific for different portions, even from a single molecule. So if there is a portion of one molecule to which the immune response is directed to, it can be specific to that particular portion and may not be available for the rest of the portion of the single molecule. So the part of the antigen to which the lymphocytes react to, so they are specifically recognized by individual lymphocytes, are called epitopes or determinants. And both these terms are going to be used interchangeably. So they mean the exact same thing, but at some points we might say these are determinants versus we may use the word epitopes. So again, two names for the same thing, but really important characteristic of adaptive immune system, where the epitopes are specific even on the same antigen. So the part of the antigen to which the lymphocytes react to are called epitopes. Now, there are various clones of lymphocytes that have different specificities. So they are able to recognize and respond to particular antigen. When the lymphocyte proliferates to make the cells, lymphocytes of the same specificity, all of those lymphocytes together are called clones, one clone for each specificity. And when that clone is selected because they came across that particular antigen, that clone gets selected. So that's called clonal selection. The cells specific for that antigen then proliferate to generate thousands of progeny. So thousands of those lymphocytes with identical receptors and identical specificity. And that phenomena is called clonal expansion. So let's look at that again. What's clonal selection? So let's see, say in someone's immune system, there are several types of lymphocytes. So a precursor develops into various different types of lymphocytes, and we'll call them clones of lymphocytes, right? So they are all shown in different colors. These are produced from lymphocyte precursors, and they are released into circulation. Say a virus comes along and the antigen on that particular virus. So one part of that antigen on that virus is called an epitope, and that might be specific for one particular clone of lymphocyte, right? So if that clone, that particular lymphocyte can attach to an antigen X, so it's specific to that particular antigen X where it attaches to it, it can now we can now say that that lymphocyte got activated so that it's going to proliferate. And it's going to proliferate to produce several lymphocytes with the same specificity, right? The purple lymphocytes are the ones that are going to be produced. The rest of the lymphocyte types or clones are not going to proliferate. So this particular lymphocyte clone got selected and to proliferate. And once it proliferates, it can differentiate to form cells, or differentiate into effector molecules, so that it can produce, say, if it's a B lymphocyte, it will produce antibodies. If it's a T lymphocyte, it can make helper T subsets or CTLs that can fight that particular antigen. So the immune response is very specific to that particular antigen. So the clone has to be selected so that it proliferates and produces the weapons that are specific to that particular antigen. So the next one is diversity. So the ability to recognize a large number of antigens due to the variability in the structures of the antigen binding sites of the lymphocyte receptors for antigens is called diversity. So the diversity has to be pretty large so that they are capable of interacting with various antigens in nature. But there are clones of each type of lymphocyte with each specificity that have, and all these clones have different antigen binding receptors. So overall, the total number of antigenic specificities of the lymphocytes in an individual can be pretty large, and it's called lymphocyte repertoire. And the repertoire can vary from one person to another, but typically is between 10 to 1,000 million types of lymphocytes or specificities of lymphocytes that might be produced in one particular individual. So the lymphocyte repertoire normally should be pretty large, and that speaks to the diversity of the antigens that these lymphocytes can bind to. Adaptive immune system has a memory. So the exposure of the immune system to a foreign antigen increases its ability to respond again to the same antigen. The first response is called a primary response, but once it forms that memory from the primary response, the next immune response is called secondary immune response. This one is more rapid, it's greater in magnitude, and it's qualitatively different from the primary response. And the memory is due to the long-lived memory cells, which are more efficient. They are already prepared, and they had some of the knowledge about that microbe or antigen in concern, so that it produces that response pretty quickly, a higher response, and say the B cells have already made class switched antibodies. They don't have to go through the usual process, but they will directly make the class of antibodies that are needed in that infection. So these cells are pretty efficient. So overall, if you look at this picture, it will show how in the primary response, say there is an antigen X, that antigen X can bind to the purple lymphocyte. It will produce that primary response and leave some memory behind. If the same antigen comes, the lymphocytes come across the same antigen, then there is a secondary response to that particular antigen, but it is specific to that antigen only, where it produces a quicker and a better response, higher in magnitude, and is more efficient. But if there is another antigen that comes along that has never been seen by those lymphocytes, that antigen will still produce that primary response and takes several weeks before it can be produced. All right. Next feature is non-reactivity to self. So this is the ability to recognize, respond to, and eliminate non-self antigens, but then not react to the self antigens. The immunological unresponsiveness to self antigens is due to various mechanisms, and it is called tolerance. So the immune system becomes tolerant to self antigens. And the reason for the tolerance is because the immune system, while it's developing, it eliminates self-reactive lymphocytes. It will inactivate the self-reactive lymphocytes that are left behind. And if there are some that still get into circulation, there is a way to suppress those self-reactive lymphocytes by regulatory cells. So that those mechanisms all together are called tolerance. All right. And then once we have looked at these features, we want to talk about the overall response to the microbes with early innate immune response and then late adaptive immune response. So let's see how that works. So the early immune response to microbes starts with the first line of defense, which is the physical barrier. That's in skin, GI tract, respiratory tract, anywhere that the body comes in contact with the outside world. This is provided by the continuous epithelium, that's the physical barrier, and then the chemical barrier also plays a role, which is provided by antimicrobial chemicals. Then there are cells of the innate immune system that are also present either in the physical barrier or inside the body where they might come across the microbes once they break that barrier. The immunity is provided in terms of inflammation and antiviral defense. So these are two mechanisms that the innate immune system provides immune response in. It causes inflammation and it causes antiviral defense. So what produces that inflammation? So there are various cells like dendritic cells, macrophages, mast cells that are present underneath the barrier and if they are watching out for any invaders. If there is an invader that they see, they are going to produce cytokines and alert the rest of the immune system to show that there is a problem. They are going to invite other cells to come and help in their fight. So the phagocytes such as neutrophils or monocytes are recruited from the blood and come to the site of infection. When these cells come to the site of infection, they again produce that immune response, and all of that together produces the phenomena that we see clinically, which is the inflammation. Also, if there is a viral infection, some of the cells of the innate immunity help in providing that antiviral defense. Dendritic cells are known to produce a cytokine called interferons. And these interferons produce what we call a cytokine-mediated reaction in form of the antiviral defense. Natural killer cells use some of these cytokines to be activated, and they kill the virus-infected cells. So that's the cellular part of the innate immunity that we see in form of inflammation and antiviral defense. There's also help from these proteins called complement and other proteins such as toll-like receptors in the blood that also help in fighting some of the providing that early response and fighting the microbes initially in that fight. When we talk about the adaptive immune response to microbes, there are various stages of the immune response. So think about the initial step where there is antigen recognition. So this is initially when that antigen enters the body and comes across the lymphocytes. There is recognition of those antigens. And once those are recognized, these lymphocytes undergo clonal selection and then clonal expansion that we talked about. So because these lymphocytes were able to recognize the antigen, they got selected to proliferate and make a clone by clonal expansion. Once that clone is produced, it is going to undergo differentiation, right? That lymphocyte activation led to differentiation so that they produce effector molecules. Effector molecules go ahead and eliminate the antigen, whether it's antibodies, other T lymphocytes that help in eliminating the antigen. And once the antigen is eliminated, there is contraction of the immune response. The antigen level goes down, so the immune response also goes down. So most of the lymphocytes undergo apoptosis, but there are few that are left behind, giving the memory, which are the surviving memory lymphocytes that survive for longer and give that secondary immune response in face of a repeat infection by the same antigen. So those are the phases: antigen recognition, lymphocyte activation, antigen elimination, contraction, and memory are the phases of adaptive immune response. One part of the immune response that's really important is cytokines. So cytokines are secreted proteins with diverse structures and functions. They help in regulating and coordinating the activities of the cells of both the innate and adaptive immune systems. They also help in growth and differentiation of immune cells. They help in activation of effector functions of lymphocytes and other cells like phagocytes, and they help in regulating cell migration and movement. And the particular cytokines that help in cell migration and movement are called chemokines. So again, really important feature of system, and it plays a vital role in providing the immune response. Um, these molecules are called cytokines that play a major role both in innate and adaptive immune system. All right, so that's the end of the chapter. So let's go through the post-test questions. Which of the following is a distinct feature of innate immunity?
Should be non-reactivity to self, C.
That's correct. So it's C, diversity, memory, specificity, way better in the adaptive immune system, non-reactive self is present in both. Which of the statements is true? Phagocytosed microbes are killed by antibodies. Intracellular microbes are killed by humoral immunity. Antibodies can eliminate the reservoirs of viral infections, and extracellular microbes may need cell-mediated and humoral immunity.
Um, isn't it D?
That's correct. So phagocytosed microbes are killed by the phagocytes. Intracellular microbes are killed by cytotoxic T cells. Antibodies are unable to reach the intracellular compartment, so they cannot reach the reservoirs. But the extracellular microbes will be dealt with by the B cells, humoral immunity, as well as helper T cells. So D is the correct answer. All right. We know that the next talk will be on chapter 2, cells and tissues of the immune system. That chapter is longer than the first one, was definitely simpler and easier to read. So the chapter 2 has a lot of other details of the anatomy of the immune system. So we'll be talking about that in next lecture. But we do have some time.